Well, we've reached the end of the road in Chem 234, and we are going to end our discussion with Experiment 9, which deals with chemiluminescence and the synthesis of luminol. So photochemistry is a branch of chemistry that studies the effect of light on chemical processes. Photochemical processes can occur in nature, and these can occur in some animals and plants. There are a few examples here. And when it is naturally occurring, we refer to this as bioluminescence. And when scientists can harness this idea of photochemistry and bioluminescence, we can use it for unique biological tools, and we call that fluorescence. So we have some goals with our luminol synthesis. Let's take a look at what the structures are going to look like that we are working with. So we'll start out by drawing our first starting material, which is 3-nitrophthalic acid. So here's the structure for 3-nitrophthalic acid. We're going to mix this with hydrazine. And we're going to use a solvent that is called triethylene glycol. So the structure for triethylene glycol is right here. So we're going to take our 3-nitrophthalic acid, hydrazine, triethylene glycol, and we're going to heat this up to approximately 210 to 215 degrees. And when we do this, we are going to generate a product where we've essentially taken both of the carboxylic acids and we've performed a condensation with the amines of our hydrazine. So it almost looks like a diamide. And this is called 5-nitrophthalhydrazide. So that is our intermediate. Then we are going to take that intermediate and react that with sodium hydrosulfite. And the formula for that, Na2S2O4. We're also going to add sodium hydroxide and water. Heat this up to about 100 degrees Celsius. And when we do that, we generate a product where we've converted our nitro group into an ammonium salt. Now I'm going to draw the product for this process as a tautomer. We're going to talk about the tautomerization of this particular product in just a second. So here is the product of our reaction. And this is 5-aminophthalhydrazide. So let's take a look at our first clicker question. So go ahead and consider what type of general reaction is occurring when we convert 5-nitrophthalhydrazide to 5-aminophthalhydrazide. So let's look at our luminol synthesis. So we've said that we take our 3-nitrophthalic acid and hydrazine with triethylene glycol for our solvent, and we form our 5-nitrophthalhydrazide. And so here's kind of a step-by-step breakdown for what we are going to execute, or what we would do in the lab, which is to take a medium glass test tube and add our starting materials in a boiling chip, heat up the mixture to gently dissolve the solid, add in our solvent, and then carefully heat the test tube again with a Bunsen burner. We're going to add a high temperature thermometer and heat until the temperature is at about 215 for 3 to 4 minutes. Cool that test tube down to 100 degrees Celsius and then add some hot water. So one of the first things that you're instructed to do when you come into the lab is to get a beaker of deionized water and heat that up on your hot plate. So make sure that you don't forget to do that once you come in. After you've added the hot water, we want to hold the test tube under cool running water. So this doesn't mean that we want to add cool water to the test tube. We just want to run the cool water on the outside surface of the test tube. And when we do this, we are going to end up generating a yellow solid, which we want to collect by vacuum filtration using our Hirsch funnel. So let's take a look at our second step now. So we're going to take our 5-nitrophthalhydrazide and we are going to add our sodium hydrosulfite and our 3 molar sodium hydroxide. We're going to heat the solution to boiling, which will be approximately 100 degrees Celsius, for 5 minutes using our Bunsen burner. Again, we're going to cool the test tube down by using cool running water on the outside surface. And then carefully we're going to add acetic acid. We want to check the pH as we're adding that acetic acid. We want to make sure that the solution is acidic. And as we're adding our acid, we're going to have a solid that is formed. And so we want to collect that by vacuum filtration, again using our Hirsch funnel. So make sure that you do empty the trap out between each of your filtrations. So dispose of that waste properly. So earlier on I mentioned that we have a tautomer that's happening here. So what we want to do is kind of investigate the tautomerization of luminol. And so let's take a look at the mechanism of what's going on here. So I'm going to go ahead and draw our starting material. Actually, I'm going to draw the product after we've reacted our initial dicarboxylic acid with our hydrazine. So we take this intermediate here, we take our 5-nitrophthalhydrazide, and we react this with our sodium hydrosulfite with sodium hydroxide and water. Then we're going to add acetic acid, and that acetic acid is going to neutralize our solution. We said it's going to end up being slightly acidic, and that's okay. So at the end of this point, we generate our neutral luminol intermediate. So we're going to notice a couple of things about our structure here. We're going to notice that our nitro group is now an amino group. If we want to be more specific, it looks like an aniline because it's an aromatic amine. And so this structure that's shown here is the structure of neutral luminol. We can consider this again to be an intermediate of the reaction. But one thing that we might notice if we look at this structure is that we've got a couple of important features that are present. So our aniline can act as a base, and we also have protons that are at our amide nitrogens that can act as acids. So what ends up happening here is that we end up seeing a very fast acid-base reaction that occurs. And so we can think of our solvent taking an acidic proton, it's one of our NH protons, and transferring it over to our basic aniline nitrogen. And so when that happens, we end up having what looks kind of like an ammonium species there. We can fill in our lone pairs. You know, I usually like to do this, so let's go back and fill those in. We don't necessarily have to, but it can be helpful for us to keep track of our formal charges. So here, we've got this intermediate. So our solvent has taken essentially a proton, an acidic proton from what looks like our amide, and transfer that over to our basic aniline. Now, this isn't going to happen twice because we only have one basic aniline nitrogen. So this acid-base reaction here is actually quite favorable in the forward direction and very fast. Now, I've kind of alluded to the fact that we have a resonance structure that we can draw for this intermediate. So let's go ahead and see what that looks like. So if we consider having that lone pair on our nitrogen, and then coming up to our oxygen, again, we can fill in the lone pairs on that oxygen if it helps us. Our oxygen is then going to have 3 lone pairs and a negative charge. We've now got a double bond between our carbon and our nitrogen. Nitrogen has a lone pair and it is neutral. So we've got these two different resonance forms of what we call our neutral luminol, because we are still overall neutral. But we call this our zwitterionic form. So zwitterions might sound familiar from biochemistry and amino acids. So here we've got our zwitterionic form of neutral luminol. Now when we add in our sodium hydroxide, we add in at least 2 equivalents of sodium hydroxide, and this is aqueous, so it's going to be in water. We end up generating basic luminol. And basic luminol is a really important intermediate for us to consider. So our sodium hydroxide is basically, that was not meant to be a pun, but turned out to be one anyway. It is going to deprotonate our amide nitrogen. Let's see if we can highlight that. It's going to deprotonate our amide nitrogen that's over here, and also our ammonium nitrogen up here. So that's where our 2 equivalents of sodium hydroxide are coming into play. And so you'll notice that I'm drawing a resonance form here where we've got our negative charges on oxygen and we've got neutral nitrogen atoms for each of our amide type species. So notice which resonance forms I've decided to draw there. Now we know that major resonance contributors, are going to most likely have our negative charge on the more electronegative atom. So this is why I've drawn the resonance form that I have here. And so this is our basic luminol. So here is kind of a summary of that process, that tautomerization process, where we take our neutral luminol and we convert that into the zwitterionic form of our neutral luminol, adding in the 2 equivalents, right? 2 equivalents here of sodium hydroxide. Again, that's aqueous. We end up generating our dianionic form, which has multiple resonance contributors. So our major resonance contributor, again, is going to be this one that's over here. So here is our second clicker question. Now we haven't looked at this particular mechanism just yet, but we will on the next slide. Let's just get to thinking about what type of reaction is occurring in the step that's shown here. So take a minute and think that one over. All right, let's take a look at the chemiluminescence of luminol. Let's take a look at the mechanism for this process. So I've drawn our major resonance contributor for our structure here of our dianionic form of luminol. And so at this point in time, we are going to add molecular oxygen, so O2, which we're generating in situ using a metal catalyst and hydrogen peroxide. So the process that's going to take place here looks very similar to a Diels-Alder reaction, which is a 4+2 cycloaddition process. So I'm going to write here that this is like a Diels-Alder. And if you'd like to review that, I definitely suggest taking a look at your textbook. And so let's see what the mechanistic arrows look like for that. So we can think about there being some electron density in the double bond between carbon and nitrogen here. And so those electrons can form a bond to our oxygen atom. The oxygen-oxygen pi bond is going to break, and it's actually going to use those electrons to form a bond to our carbon atom. And then the double bond electrons that are between our carbon and our nitrogen are going to shift to form a double bond between nitrogen and nitrogen. So this process, lots of arrows, it's a concerted process, 4+2 cycloaddition. And gives us a product that is very interesting looking. So go ahead and fill all of that in. So we now have a double bond between our 2 nitrogen atoms. We have single bonds between our nitrogen and carbon atoms. But now we've attached basically an oxygen atom to each of our carbons. fill in all of our lone pairs just to be thorough here. And so we've got this really interesting intermediate. It kind of looks a little bit odd. But we know that carbonyls, so carbon-oxygen double bonds, are very thermodynamically stable. And so having these oxygen atoms bonded to each other, we'll highlight that, having two oxygen atoms bonded to each other is quite unstable. So an oxygen-oxygen bond is not really a very stable bond to have. So what's going to end up happening is that this intermediate is going to decompose, and we're going to release N2. We're going to release nitrogen gas. And so we're going to reform the carbonyl at this position. Then we're going to see how our N2 is going to form. So the electrons between carbon and nitrogen are going to go to form a triple bond between our two nitrogen atoms. Then our carbon-nitrogen bond here is going to break and become a double bond between carbon and oxygen. And then finally, what we were after from the beginning was to get that oxygen-oxygen bond to break. And so this step here is going to be essentially irreversible. And it's irreversible because we are generating N2 gas. So we're generating that nitrogen gas. And once that gas is generated, it will bubble out of solution. And we can't go in the backward direction. So our N2 gas, we'll put our arrow there to show that that is going to be released. Now this is our excited state. And so chemiluminescence is going to occur when we are relaxing this molecule from an excited state into a ground state. So the excited state is going to relax and then we are going to end up with our ground state plus light. And that process is our chemiluminescence process. And so the two structures, the chemical structures of these look the same. We represent that they are different by putting our excited state in brackets with a star. And then we have our ground state here, which we do not put in brackets. And here we're also generating light, and so we're going to express that by labeling it H nu. And that's what's going to cause our luminol to glow. So this mechanism, there's a lot of stuff that's going on. Our 4+2 cycloaddition, that's a little bit tough. This decomposition mechanism, which, we generate our excited state plus our nitrogen gas. That's a lot of arrows that are going on. Just keep in mind that the driving force for this, we're generating N2 gas, which is very, very thermodynamically stable. We're breaking that oxygen-oxygen bond, which is very weak. So this is going to be a really favorable process. And so luminol is used a lot, and we've seen it all on TV if you've ever watched an episode of CSI. And so the oxidation of luminol under alkaline conditions, under basic conditions, produces light. And so the iron in hemoglobin in blood can act as a catalyst for the oxidation reaction. So if you remember when we talked about that reaction between our, let's see if we can go back and revisit, between our dye anion. In our O2, we have a metal catalyst here, and so iron in our blood can act as that metal catalyst. And so the faster the rate, the more light that is produced during this reaction. And so that energy is given off as light. So chemiluminescence, we consider chemiluminescence to be a process in which a molecule undergoes a chemical reaction that generates a product in an excited state. That excited state will then emit light when it returns to the ground state. And so here is our molecule that's our starting material. It's undergoing some chemical reaction, which is represented by our arrow right here. And it's being converted into an excited state molecule. So it has undergone a reaction, that's why it's not A going to A star, it's A going to B star. The structure has changed. It is a different structure and an excited state structure, which then relaxes. So here's our relaxation step. And that is going to generate B, which is, again, the same structure, just is no longer in our excited state. So our ground state is here of B. And we've also generated light. And so there's an energy kind of diagram down here that expresses that. So we can go ahead and we can summarize this process. So a molecule undergoes a chemical reaction. Generating an excited state product. So this excited state product will emit light upon returning to the ground state. So there's kind of our general summary of chemiluminescence. So we can take a look at this process from an energy perspective. We can look at our rough enthalpy calculation going from, let's call our dye anion A to our final product over here. So if we look at the overall bond energies, we can see what this calculation looks like. And so take a minute to look that over, make sure that you understand which bonds are being expressed here as being broken, going from our conversion of A to B. We're not looking at any of our intermediates here in the middle, so we're kind of ignoring because they are intermediates, so we just want to focus on the difference in energy between A and B. Which leads us to our next clicker question. So if we consider the overall energy perspective on the last slide, is the reaction energetically favorable as it's written? So going from our dye anion to our product, is it favorable as written? So chemiluminescence is particularly interesting, but it would be really great, right, if we could change the colors and we can generate different colors. So if you have chemiluminescence that always gives you the same green color, that's great, but you might want to be able to probe different processes. So you might want to be able to generate different colors. And fortunately, we can do this. And we do this with the help of dyes. So adding dyes lets us change the color. And so we can think back to when we were talking about azo dyes. We talked about conjugated systems, how conjugated systems can absorb energy. When they relax, we end up seeing some interesting types of processes. And so here, to talk it through, we're again starting with our starting material A, which is undergoing a reaction. And that chemical reaction is producing an excited state product that is chemically different than our starting material, and it is in the excited state. Now at this point, we can add in some dye, and that dye will then absorb energy from our excited state, B. And so now what we've got is we've got excited state dye, and we've also got B, which has relaxed. So we've had this sort of energy transfer between the excited state of B and our dye, which was in the ground state and is now excited. Then C, our excited state dye, is going to relax. So C relaxes. And that gives us our ground state of the dye plus light. So here are a couple of structures of some energy transfer dyes that we can use to look at this process, and you will see in the video for this experiment that we can use this structure that's on the left here, eosin Y. And when we do that, we get a different color that is given to us upon our chemiluminescence. And this is going to be a unique, distinct color that is the result of the electron density of the dye. So we've got highly conjugated systems here that again can absorb that energy. With that, we have wrapped up our experiment 9, our last experiment for Chem 234.